Well, the world of amino acids, peptides, and proteins begins with an alphabet of 20 amino acids that your body uses to make proteins, to make the enzymes that are doing the chemistry, to make the proteins in your muscle cells that allow movement, to make the proteins on the surface of your cells that allow them to communicate with their environments. Proteins are the real workhorses of biology. And there's a huge amount of diversity that you get in protein function from a simple alphabet of just 20 amino acids. In much the same way that there is a huge diversity of words and sentences and paragraphs and books that you can get from 26 letters in our alphabet. And actually the analogy of amino acids as being letters or words and proteins as being sentences or paragraphs is actually pretty decent. And we've got a project going on in my lab where students are trying to use an AI model that's designed to read English and seeing if it can read amino acid sequences and make interesting predictions. Other people have done it before. We're working on a small area of it. But just like when you were learning to read, you had to first learn to recognize letters. If you want to understand proteins, you need to recognize amino acids. And so we're going to be learning the 20 amino acids. By the time you do the problem set or take the exam, if I write a string of 10 letters, you need to be able to draw the peptide that's represented by those 10 letters. And so it's painful, but it also gives you the tools to understand biology at the atomic level. You want to know what the atoms are doing. And this is maybe the only class you'll ever take that will give you this information, so it's worth knowing. And the reason it's worth knowing is that in biology, even a single amino acid difference can sometimes make a huge difference in the function of the protein. And so you want to be aware of those things. All right, so our first task is to understand the 20 amino acids. The basic structure of the amino acids that your body uses to make proteins is as follows. They are called alpha amino acids, and the thing that unifies them all is that they all have a carboxylate group. They all have an amino group at physiological pH. I'll draw it in its fully acidic form. At physiological pH, the pKa of this carboxylate, we've said it's basically 4 all semester. It turns out that the pKa of 4 is for a carboxylic acid with no other electronegative atoms nearby. For example, the pKa of acetic acid is around 4. It turns out that the pKa of this carboxylic acid is a little bit lower than you would expect because of the presence of the nearby electronegative nitrogen that tends to pull electron density away from the carboxylate, making the conjugate base more stable and making the proton not held on as tightly. This isn't really a major point, but the pKa of a carboxylate in an amino acid is around 2. At physiological pH, it doesn't make any difference at all, right? At pH 7, the carboxylic acid is going to be in its neutral form, whether the pKa is 4, the carboxylic acid is going to be in its negatively charged form, whether the pKa is 4 or 2. The pKa of this ammonium group is also a little bit lower than expected for a typical ammonium group. It ends up being around 9, and that's because of the presence of the nearby electronegative oxygens that pull electron density away from the nitrogen and make it stabilize the conjugate base. So that's the basic structure. You have a carbonyl carbon, you have an alpha carbon, and then you have a nitrogen. The nitrogen is on the alpha carbon, which is why we call this an alpha amino acid. So if I were to ask you to draw what form this amino acid would take at pH 7, Well, the ammonium proton should still be there because its pKa is 6, but the carboxylate proton should be gone because its pKa is 2. I think we opened the door earlier today. One of my colleagues taught in here and said it was like 100 degrees in here without the door open. So we'll keep it open just in case, but if it gets too noisy out there, we'll close it. All right. Of course, if we lowered the pH below 2, then we would protonate the carboxylate. Any questions about that? So that's your amino acid. Now there are 20 of them. How do they vary? They vary in the alpha carbon. Some amino acids will have other functional groups at the alpha carbon. The one I just drew for you is called glycine. And amino acids are frequently represented by a 3-letter abbreviation, GLY, and the first letter is always capitalized, or a 1-letter abbreviation, which is just G. And we'll learn that for each of the amino acids. Glycine doesn't really have a side chain. At the alpha carbon are simply 2 carbon-hydrogen bonds. So there's no stereocenter in glycine. We'll now talk about the remaining amino acids, and as we do, I'm going to introduce them to you in groups because you'll see that some of the amino acids have similar properties. And some of what we talk about now will be useful to us Friday or Monday when we start talking about protein structure. All right, any questions about basic amino acid structure? One other word that you may have heard, a molecule that is neutral but still has charges in it, 1 plus and 1 minus. I don't anticipate myself ever asking you a question about this, but you might hear this word. It's zwitterion, or if you want to pronounce the W like you would if you were saying this in Germany, Zwitterion, some people say, but anyway. It's a neutral molecule that has a plus and a minus that cancel each other out. Notably, the neutral form of the amino acid is not the main form that exists at physiological pH. So you can draw amino acids that way, and when you work with them as a synthetic organic chemist, they're often this way in organic solvent, but they're this way in biology. Okay, so let's introduce the team to you. The first one is called alanine. This is maybe the simplest one. Its 3-letter code is ALA, and its 1-letter code is A. All of the amino acids are going to have the same, what I will call backbone. The backbone is the carbonyl carbon, the alpha carbon, and the nitrogen. But they will differ in the side chain. For alanine, the side chain is a methyl group. And there is, I will, implied but not shown, is a proton that's coming up and pointing out at you. So this is alanine, and I will call it basically a generic amino acid in the sense that it's not polar, but it's also not that hydrophobic. The side chain is small, and the side chain is not charged. So I'm calling it generic because alanine is comfortable in a lot of different environments. Alanine has a stereocenter. The stereocenter is here at the alpha carbon. If you assigned the stereochemical configuration of this stereocenter, it would be S. Nitrogen is priority group 1, the carbonyl carbon is 2, the methyl group is 3, 1, 2, 3, it's clockwise, but proton is up, so it should be S. The stereocenters at the alpha are always at the alpha, well, the stereocenters at the alpha carbon for the amino acids are always going to be S, except for an amino acid that's called cysteine, where a sulfur in the side chain causes it to be R based on a priority change. But they will all have this same basic shape. In other words, if you draw an amino acid with the nitrogen on the left and the carboxylate on the right, and the alpha carbon pointing downward at sort of a valley at the V position, the side chain will always be a dash. If I rearrange things and drew the molecule like this, with the nitrogen on the left and the carboxylate on the right, but the alpha carbon at sort of a peak, then the side chain would be a wedge rather than a dash. And of course, if I, and you could rotate things around to see other views as well, but it's important that you get the stereochemistry right. Amino acids that your biology uses, these 20 amino acids that biology uses in proteins, you will sometimes hear people talk about them being L amino acids. That refers to the stereochemistry at the alpha carbon. And that's, as I've said before, an old-timey designation about chirality. Interestingly, most carbohydrates that your body uses are all called D carbohydrates. Most amino acids your body uses are all called L. And we don't need to delve too much into that. But for you to get the structure correct, you must have the side chain chirality accurate, okay? So if someone writes ALA or capital A, they mean the amino acid with this chirality. They don't mean the enantiomer. Okay, questions about that? So next we'll do some amino acids that I will generally describe as nonpolar. And these are mostly saturated hydrocarbons as the side chain. All right, so the first is valine. Three-letter code VAL, one-letter code V. So far, the one-letter codes make a lot of sense. We'll get to the point where we run out of letters and they stop making sense. Valine's got the same backbone structure. At the alpha carbon, valine also has a wedge. And then at the beta carbon, you've got two branching methyl groups. So valine is, all of the amino acids in this category, nonpolar, are going to be somewhat hydrophobic, meaning that there will be some preference for them to not be exposed to water. Another interesting thing about valine is it's got a branch here at the beta carbon. You got a place where the side chain goes in two different directions. We'll call this a beta branched amino acid, a beta branched side chain. And this idea of a branched chain amino acid shows up in a metabolism of amino acids, and it also shows up in marketing information. For example, you can buy protein powders where they say these are rich in branched chain amino acids or BCAAs. You heard that before? Okay. The reason that's a thing, as you will see, is the metabolic fate as they're degraded of the branched chain amino acids is a little bit different than the metabolic fate of amino acids that don't have branched chains. And we'll talk about that maybe a little bit more next time. All right. The next two are also branched chain amino acids. The first is leucine, Leucine, 3-letter code LEU, and 1-letter code L. Again, that one makes sense. You've got the same backbone and the same side chain chirality. At the beta carbon, though, there's no branch point. You go to another carbon, we'll call that the gamma carbon. And finally, at the gamma carbon, there's a branch point. So leucine's like valine if you inserted a CH2 in between the alpha carbon and the beta carbon. And this is also, leucine is also hydrophobic and also is also a branched chain amino acid, though it is gamma branched rather than beta branched, meaning the branch point's on the gamma carbon. Okay, another one is an isomer of leucine, and so it's unsurprisingly called isoleucine. And this one's going to be tricky because isoleucine has 2 stereocenters, and I always forget. So let me show you the basic backbone structure of isoleucine. Same as with all the other amino acids. Dash here. Isoleucine is beta branched like valine. And I'm going to get, we'll just draw it like this. So here's the alpha carbon. At the beta carbon, you have a branch point. Only one of the 2 branch points is a methyl group, and the other is a CH2 that's bonded to a methyl. It's like you took a CH3 from the gamma carbon on leucine and slid it over to the beta carbon. So isoleucine is an isomer of leucine. Its 3-letter code is ILE, its 1-letter code is I. Like leucine, it's hydrophobic. Unlike leucine, the branch point is at the beta carbon. The stereocenter at the alpha carbon in isoleucine, as with the other alpha L amino acids, is S. For regular isoleucine, let's see. The other stereocenter should also be S. And I think I've drawn it wrong here. So we'll put the other methyl group there. This stereocenter should also be S. Let me just check on that because I want to make sure that I don't tell you the wrong thing. But I'll assure you that I'm actually not going to test you on the side chain stereocenter for isoleucine, okay? Because if I've been studying peptides and proteins for 20 years and I don't know it, you probably don't need to know it either. Sorry, it's taken me a minute to figure it out. Should have figured this out before. Yep, I was right. They're both S. Okay, sorry about that. I do want you to know, absolutely, the stereochemical configuration of the alpha carbons. you're going to see that there's another amino acid like isoleucine that has a stereocenter in the side chain. And like isoleucine, we're not going to care about the configuration. The way you're going to recognize isoleucine is just its beta carbon has a CH3 here and an ethyl group over here. Okay, any questions about isoleucine? Turns out if you look at those two, you're like, what's the big deal? You just slide a methyl group over. I could tell you a very interesting story about how that switch changes whether something folds one way or another way, but we probably won't have time for it. But those kinds of subtle structural changes can matter. Like valine, isoleucine is a branched chain amino acid, and its metabolism is a little bit different from amino acids that don't have branched side chains. Okay. So there's one more that we're going to put in this nonpolar category, and that last one is methionine, METIONINE, 3-letter code MET, 1-letter code M. Methionine's a little bit weird. It's nonpolar, but it's not exclusively hydrocarbon. So alpha, beta, so this is the methionine side chain. Alpha beta, alpha carbon, beta carbon, gamma carbon, and then on the gamma carbon there is a sulfur, and that sulfur is bonded to a methyl group. This functional group is a weird one. You haven't seen it before. That's called a thioether. Overall, methionine is relatively hydrophobic like the other nonpolar amino acids, though it turns out this thioether can do some interesting chemistry that we won't talk about for the most part. Methionine is the amino acid encoded by the start codon in mRNA, and so in proteins that you express in bacteria, methionine is always the first amino acid in the sequence. Okay, so those are the nonpolar amino acids. Names, 1-letter codes, structures, and then know something about their overall character. That is, these are all nonpolar with saturated hydrocarbon side chains. They're all relatively hydrophobic. The stuff about the branching is mostly to make you familiar with terms that you might encounter when somebody's trying to market something to you. Though if you know the structure, answering the question which ones are branched is trivial. If you know the structures, well, there's branch points at the beta or gamma carbons. You can figure that out. Okay, there are questions about what I want you to know? Yeah?
Sorry, what's the word after start?
Start codon, C-O-D-O-N. We'll talk about what that is when we talk about transcription or translation. But that's a 3-letter sequence of nucleotides that indicate start making a protein and start reading here. Okay, other questions? All right, let's keep going. The next category of amino acids we're gonna call aromatic amino acids. And aromaticity, or being aromatic, is something that I haven't taught you about, but it has to do with having unsaturated cyclic hydrocarbons. It turns out there are some arrangements of atoms that are particularly stable. An example of something that is aromatic and very stable is a benzene ring. 6 carbons in a 6-membered ring with double bonds between each of the carbons, alternating double bonds. Benzene is an example of something that is especially stable, even more stable than an alkene, and that special stability is called aromaticity. You hear the word aromatic and you probably read the word aroma and think, oh, these compounds must have a characteristic smell. Benzene does have a characteristic smell. It's not particularly like, ooh, I love that aroma. And molecules that are aromatic sometimes don't have any particular smell. That's an old word that was used originally to characterize some molecules that did have a particular smell and had these types of rings in them. But in any case, what you're looking for aromatic amino acids is amino acids that have some kind of benzene ring or some kind of ring that has double bonds in it. So the easiest of these is just, sorry, we start with the name, don't we? The easiest of these is called phenylalanine. Its three-letter code is PHE, and its one-letter code, we're going to use the letter P for something else, and in any case, the letter P doesn't capture the F sound for phenylalanine, so we're going to use the letter F. Makes decent amount of sense. Here's the backbone. On the alpha carbon, you have a dashed the beta carbon. The beta carbon will then be attached to a benzene ring, which is sometimes called a phenyl group. So now the name phenylalanine makes sense. It's like alanine. It has a methyl group, except one of the hydrogens on alanine has been replaced by a benzene ring. The characteristic here is that phenylalanine is nonpolar and fairly hydrophobic. There's something else really, really cool that I want to tell you about benzene rings that would be useless to you in this class. So I'm going to stop myself. But if you want to know more, come talk to me. There's something special that benzene rings can do that doesn't exactly fit in with being hydrophobic and nonpolar. Okay, so that's phenylalanine. A close cousin of phenylalanine is the amino acid tyrosine, Tyrosine. Its three-letter code is TYR. But its one-letter code, we're going to use T for something else later, so we use Y. So this is BYU's favorite amino acid, because there's no amino acid with the letter B or the letter U, so this is all we've got. Tyrosine looks just like phenylalanine, only it's got, light bulb. Can we get the bookstore to offer a Reppin' the Y t-shirt that has tyrosine structure on it? Reppin' the, and then just tyrosine? Can we propose that? I would buy it. I would definitely buy it. Okay, so it's like phenylalanine, only it's got an OH group. So tyrosine, you have to be, and the OH group is opposite the beta carbon part of the side chain. Tyrosine is difficult to describe. It is mostly nonpolar. It is mostly hydrophobic, but the OH group is polar and is capable of hydrogen bonding. We learned about that, what now seems like months ago. And the other interesting thing is this OH group can be deprotonated. The proton on this OH group has a pKa of 10. That's lower than expected for an alcohol. And the reason is the conjugate base, which I'm just going to draw the side chain portion, the alkoxide, is stabilized by resonance. So there are times in biology where this molecule loses its proton and becomes negatively charged. And I'll show you just one of the resonance structures. This is one of them. But if you followed the pattern, you would discover that not only the atoms that I've shown can share the negative charge, but also the other ones that I just highlighted. Okay, so tyrosine is different than phenylalanine. It has a side chain that can be deprotonated. There are some biological reactions you won't encounter where that oxygen acts as a nucleophile. Importantly, tyrosine is a precursor to another amino acid that's not used in proteins called L-DOPA, and L-DOPA is a precursor to dopamine, and we're actually going to talk about dopamine synthesis probably next time. Okay, questions about that? Okay, I feel like I'm introducing you to my old friends. This one is tryptophan. You've probably heard about that before because everybody that serves turkey at Thanksgiving is going to comment afterwards, oh, I ate a lot of turkey. It's making me sleepy. It must be the tryptophan. You probably heard that before. I don't know what the evidence for that is. Tryptophan is a precursor to the neurotransmitter serotonin. We'll talk about it being its biosynthesis next time. The three-letter code for tryptophan is TRP, trip, makes sense. And actually my PhD advisor once wrote a News and Views article about somebody else's paper, and this is common in chemistry. If somebody writes a really cool paper, somebody else will write a little bit, hey, everybody, check out this really cool paper. And the paper that had been written was about the role of the amino acid tryptophan in some peptide. And my advisor could not resist using TRP to say trip. There was some old phrase about tripping to the light fantastic, so I don't know what that means, but you'll have to Google it, look it up, I don't know. But his title was like, mini proteins trip TRP to the light fantastic. And it was punny if you were a nerd. Sometimes if you use amino acid puns, you can detect fellow nerds. The one-letter code for tryptophan is the first one that doesn't make sense. It's W. And for this, to remember this, you can pronounce tryptophan as though you were the Looney Tunes character Elmer Fudd. Do you know who I'm talking about? You don't? All right, hold on. This is not going to be in the video because it's unlicensed, but I will just Google Elmer Fudd is a Looney Tunes character. He's always trying to hunt Bugs Bunny. You know that guy? And he struggles to pronounce the letter R. And so if Elmer Fudd were saying tryptophan, he would say twiptophan, and that's a W sound. So that's how you can remember tryptophan. Okay, the structure of tryptophan is, the ring structure is hard. It's more complicated than benzene. It's got the same backbone. Like the other aromatic amino acids, it's going to have the same alpha carbon and the beta carbon. For tryptophan, though, the beta carbon is connected to a 5-membered ring, and that 5-membered ring is connected to a benzene ring. And if you looked around, there's all of the atoms in this ring are sp2 hybridized. They're part of a conjugated pi system. I think those are words we have not talked about and you don't need to worry about. You can accept it on faith that tryptophan is aromatic. Tryptophan, like tyrosine, is mostly nonpolar. It's mostly hydrophobic. The NH is capable of hydrogen bonding, but is not very acidic. So we don't even worry about the pKa of that NH group. As I've said, and maybe we'll just note here, because it'll come up tomorrow, tyrosine is a precursor to dopamine, and tryptophan is a precursor to serotonin. Now there's one last amino acid that I could include in this category of aromatic amino acids because the side chain is aromatic. However, I'm not gonna put it in this category because it's much more polar than the other amino acids. Okay. All right, so we've so far talked about amino acids that are saturated hydrocarbon nonpolar amino acids. That's V, L, I, and M. We talked about the generic amino acid alanine, which some people include in the list of nonpolar amino acids, which is fine. It doesn't have polar functional groups, but the side chain's pretty small, so alanine's not that hydrophobic. So I'm sort of including it separate. And then we've done the aromatic ones. Now let's talk about the remaining amino acids are going to be polar but neutral, polar and charged, plus or minus, and then there will be two that are just weird. If you've seen the Barbie movie, there's Weird Barbie, and these are the Weird Barbie amino acids. In the sense that you've already met one of them. Glycine lacks a side chain, so glycine is going to be in our weird category. And then there's another one called proline that's also weird, and we'll talk about why. Okay, let's do charged amino acids. These are amino acids that are charged at physiological pH. So I'm going to draw them in their charged form. Understand, if you go look up the structure of an amino acid, it might be shown in its neutral form, and you'll have to use pKa knowledge to remember this. We'll do positively charged ones first. The first one is called lysine. Its three-letter code is LYS, and its one-letter code is K. That's another one that doesn't make sense. We already used L for leucine. There's no K in the word lysine, but you got to make a call somewhere. I have been studying this for so long, as I said, 20 years now. When did I start? Yeah, I started my PhD in 03. Dang, I got old. In any case, I've known that lysine was the letter K for so long that it just seems like obvious, like it was always that way. I don't know why it's the letter K, and I understand you'll have to adjust to that. Here's lysine. It'll help if you know Greek letters to remember the lysine side chain. The biggest problem here is there's a lot of carbons in it, okay? So the way I remember lysine is alpha carbon, beta, gamma, delta, epsilon, and then an NH3 group. Delta, epsilon. So lysine is cationic. That means it has a positive charge. And the positively charged NH3 is on the epsilon carbon. That's the Greek letter epsilon. It looks like a curly E. And the pKa of this ammonium group, the pKa from going from positively charged to neutral is 11. So here's the neutral lysine side chain. At physiological pH, it's definitely going to be positively charged. There are some conditions in which lysine is neutral or close enough to neutral that you can do chemistry on this amino conjugate base. That really won't come up too much in this class, so we won't worry about it. When you see the letter K, you need to be thinking positively charged. Because of the charge, this is, we would call it, polar. We would call it hydrophilic, meaning it likes water. Okay, questions about that one? Yeah, go ahead.
What are the names of the Greek letters?
Sure, yep. Greek alphabet, let's see. Alpha, beta, gamma, delta, epsilon. All right, so the next one is also positively charged and is called arginine. Its three-letter code makes sense, ARG. So this is the favorite amino acid if you're frustrated. It's also, and so the one-letter code here makes some sense. It's R for arginine. And this is also the answer to another chemistry nerd joke. What is the pirate's favorite amino acid? Arginine. Is that going to be a question on the test? Yeah. Can it be? What is the pirate's favorite amino acid? Okay. Draw the pirates. Which of the following is the pirate's favorite amino acid?
Let's see how it's drawn first before we make that.
Oh, okay, yeah. You haven't even seen it yet. Arginine, unfortunately, is more complicated than lysine. It has a function. You've actually seen it, I think. I think there was an arginine in a peptide that I gave you on, what is the charge on this molecule type question before. You've got, for arginine, you've got the alpha, the beta, the gamma, and the delta carbons, and then attached to the delta carbon, you have a nitrogen, but it's not just a nitrogen, it's a nitrogen that's bonded to all of this with a positive formal charge on this nitrogen, but which could be delocalized on each of these other nitrogens via resonance. So arginine, I'd say, is sort of on the level of tryptophan in terms of its structural complexity. This functional group in arginine, in lysine, the functional group was called ammonium. In arginine, it's called guanidinium. That's not really anything to memorize. I just might say those words and you should know what I'm talking about. Like lysine, arginine is polar and hydrophilic. It's positively charged, and the guanidinium group is on the delta carbon. The pKa for the guanidinium group is 12. The neutral form would look like this. The neutral form would lose a proton from the positively charged nitrogen. The neutral form is called guanidine. You don't necessarily need to know that, but I might call it that. So at pH 7.4, it's always positively charged. Okay? So when you see K's and R's, you need to be thinking pluses, positive charges. Okay, questions about those ones? So in terms of, memorizing these, you should do whatever works best for you to memorize. You've had classes before where, and this isn't really one of them, you've had classes before where you've been able to succeed mostly by memorizing stuff. This class has been mostly about can you apply what you've learned, not can you repeat facts. But with the amino acids, this is a place where you're going to need to repeat facts. So if flashcards work for you, there's probably flashcard sets available somewhere on the internet. You could search a website, test me on amino acids. You could probably ask ChatGPT to do that too, and he could probably test you on it. The AI chat could probably test you on that. In any case, be careful though. Chat is not really good at chemical structures. If you ask it to draw structures, it sometimes gets them wrong. So, yeah, it'll get better, but it's not yet there. All right, let's do the negatively charged amino acids. So arginine counts as cationic as well. The first is called aspartate, or sometimes people call it aspartic acid. I call it aspartate because at physiological pH, it's in its carboxylate conjugate acid form. The three-letter code is ASP. We've clearly already used A, so we're going to use D, which makes no sense at all. So that's going to be one you got to just memorize. In aspartate and the other one I'm going to show you are really easy to mix up. Aspartate is the shorter side chain. So alpha, beta carbon, and then attached to the beta carbon is the carboxylate. So it's got the gamma carbon is the carboxylate one. The pKa of its conjugate acid is 4. So I'll just draw the conjugate acid. The pKa is 4. So at pH 7, this one should be negatively charged. It's polar, it's hydrophilic. Other than that, I think that's enough. You have seen aspartate before in the catalytic triad that is present in the lipase enzyme that hydrolyzes triglycerides. Yeah. Hand up, no?
Why does it stay negative at pH 7.4 if
pKa is 4. If pKa is for the conjugate acid, the neutral conjugate acid. That's why I've drawn the arrow in this direction. So when pH is higher than pKa, yeah, then the proton's gone. The next one is almost exactly like aspartic acid. It's called glutamate. Sometimes people call it glutamic acid. I call it glutamate because at physiological pH, it's almost always in its negatively charged form. Its 3-letter code is GLU. Its 1-letter code is E, which again doesn't make a whole lot of sense. There is an E in glutamate, but there's also one in aspartate. So what are you going to do? Yeah.
Spell blue, like the sticky stuff.
Glue. Oh, that's an interesting mnemonic device. You guys hear that? So take the 3-letter code glue and then say, oh, if I were going to spell glue, there would need an E, and then E is the 1-letter code. Thank you. I've never thought about that before. That's good. All right, so glutamate is going to be just like aspartate, only there's one more carbon in the side chain. So you're going to go alpha to beta to gamma and then the carboxylate. So in glutamate, the carboxylate is the delta carbon. Other than that, same sorts of things as aspartate. Should be negatively charged at physiological pH because its conjugate acid has a pKa of 4. So it loses its proton at physiological pH. All right, let's see how we're doing. We did 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and you've already sort of met the weird glycine. So, okay, we're more than halfway through. We'll take a brief break here. What we're going to do afterward is finish the rest of them, and then we're going to start showing you peptide structure. That is how to draw a peptide, a polymer of amino acids. And then we'll talk about protein and peptide hydrolysis by the enzyme chymotrypsin, and we'll walk through that. All right, so let's take a few minutes, and then we'll be back in about 5, yep.